Hi, welcome to the last chapter of the class, the lymphatic and immune system. This will almost certainly be two videos because the immune system is a little bit long and quite a bit complicated, but I think totally cool, so maybe you'll like it too. The lymphatic system I think is boring, but it's mercifully short. So let's get to it. First up, why is this not advancing? There we go. Um, so you just want to know the basic functions of the lymphatic system. As it says right here, it returns interstitial fluid and leaked plasma proteins back to the bloodstream. So all of your vascular capillaries, your blood capillaries, are leaky. Lots of fluid leaks out of them and some of the plasma protein. That fluid and protein needs to be returned to your circulatory system, or we would all just swell up all the time. We would have edema, which is accumulation of the interstitial fluid. And because this fluid comes from your bloodstream and also travels through your body, this ends up being the structural home of the immune system. So as lymph flows through lymphatic tissues, your immune system hangs out there and monitors the lymph and cleans up infections. So here is a little bit of anatomy. You won't see this picture on the exam, but you do need to know the drainage pattern for the body. So you have a right lymphatic duct and a thoracic duct. And as it says there, the right lymphatic duct drains upper right arm and the right side of the head and thorax. So everything in purple goes into the right lymphatic duct. Then you have the thoracic duct, which we call it the thoracic duct instead of calling it the left lymphatic duct, because it does go up through the thorax before hitting those blood vessels there. So the rest of the body drains into that. And both of them dump the blood into, as it says there, where the internal jugular and the subclavian veins meet. So it's really the subclavian vein at the junction of subclavian and internal jugular. I don't care that you know the capillaries, vessels, trunks, and ducts, it doesn't matter. What I do want you to know is that all of your lymphatic vessels, like your veins, have valves, so that there's only one-way flow for the lymph, because the pressure is very, very low in your lymphatic system, right? It is completely disconnected from your vascular system, so there's no pump pressure coming from the heart. So your lymphatic system completely relies on your respiratory and muscular pumps to get the lymph moving through your lymphatic vessels. There are two things that I want you to know about lymphatic capillaries. One is that they are very permeable. Part of how they end up being permeable, and this picture doesn't really do it justice, if I zoom in, right, there's a little bit of overlap between those two cells right there. Oh, they have the flap going the wrong way. That's so silly. So this cell here can flap in and let fluid flow in, but it is attached to connective tissue by fibers that prevents it from flapping out. So it's kind of like a carabiner where you can push it in and put your keys on it or carry a water bottle on it, but it has a very strong attachment. You can't open it the other way. That's the way these endothelial flaps work. Whoops, that all got crazy. So it's very easy for fluid to get in, harder for fluid to get out. Again, because of the low pressure, you don't want any backflow. So one of the consequences of this is that it is very easy for cells like cancer cells to get into your lymphatic system. So cancer cells can break off from the tumor, invade blood vessels, and circulate in the blood, but because it's so easy to get into a lymphatic capillary, the cancer travels quite often through the lymphatics, which is why, especially like with breast cancer, usually the first place it metastasizes to is the lymph nodes in the armpit or axillary region. I also just want to make you aware of these special lymphatic capillaries that are found in the intestines called the lacteals, and that is where your dietary fats get absorbed. So lymphatic tissue or lymphoid organs, you should know, are made out of reticular tissue. You may remember this from Anatomy and Physiology 1. It has this branching network of what is actually collagen, but it is arranged in such a way, the architecture of it, when collagen is arranged in this physical form, we call it reticular fibers. So it's this branching network that provides a framework or a scaffolding for all of the cells. So if you look at all these little purple circles, those are nuclei. That's a whole lot of cells packed closely together. And then what happens is that this acts kind of like a filter, and fluid trickles through and past all of these cells, and you have cells like macrophages here that are going to physically cleanse the blood, right? They phagocytize debris that's not supposed to be in there. And we haven't gone over lymphocytes yet, these guys, but they are part of your immune system. So they are white blood cells that are part of your adaptive immune responses. So that is reticular tissue. You want to think of it as basically like a Brita filter, where the water trickles over all the charcoal in the filter, only instead of all the charcoal being in a bag, it's all connected together like bunches of grapes on a grapevine. So this is what the reticular tissue looks like. I include the pictures even though they're not on the exam, but each one of these little light areas is called a germinal center, and that marks the middle of a follicle or nodule. So every time you see, I have to remember not to move the mouse too much because I know it gets annoying when you're watching it. One of these light centers here, here, or down here, you can kind of see there's a light bit, there's a light part, there's a light part. You get used to spotting lymphatic tissue. And unfortunately, we're almost done with the class, so you're not going to have time to look at other organs and find the lymphatic tissue in those organs. Lymphocytes for now, we're not going to spend a lot of time on because we have the whole next chapter coming up. For now, just know that T cells we are going to associate with viruses, and B cells we are going to associate with antibodies. And both of these cells actually circulate continuously. So any lymphocyte, if we go back here, right, will spend a little bit of time attached to these reticular fibers hanging out in a lymph node or the spleen or a tonsil. Then it'll let go and get flushed through with the lymph and end up in the bloodstream, and then it might hang out somewhere else in a different lymph node or in a different organ. So your immune cells spend a little bit of time in each part of the body. This is why your immunity is systemic. And then just making sure I'm not getting mixed up with the slides. So then what we're going to do is go through individual lymphoid tissues, and there are two main types. You have lymphoid organs. So these are chunks of lymphoid tissue that are discrete and separate from the organs around them. They have their own capsule or membrane that surrounds them. Then you have what is called diffuse lymphatic tissue. The most prominent example of is called MALT, mucosa-associated lymphoid tissue. Here is a little lymphoid nodule inside of somebody's lung. So it's not a separate tissue or a separate organ. It is lymphatic tissue found within another organ. So we're just going to run through these quickly, and you basically want to know the function of each lymphatic organ or the location of the different kinds of diffuse lymphatic tissue. So first up is the lymph nodes. They are located along the route that lymph takes from lymphatic capillaries through vessels and trunks back into the bloodstream. So all lymph will pass through a lymph node at some point before it gets dumped back into the bloodstream. There are three major superficial accumulations of lymph nodes. So you have a lot of lymph nodes in your inguinal region, your axillary region, and your cervical region, right? That's if you go into the doctor and you have a sore throat and she or he is feeling your throat, they're feeling for lymph nodes. People usually say swollen glands. But they're not checking to see if your salivary glands are swollen. They're checking to see if your lymph nodes are a little bit inflamed, because that's where your white blood cells are, and when you have an infection, you get mitosis or proliferation of your white blood cells. Moving on. Oh yeah, so the function of lymph nodes, not to be confused with the lymphatic system, because they're very similar, right? So the lymphatic system is returning lymph to the bloodstream and housing your immune system. Within that system, the lymph nodes are responsible for filtering the lymph, and this is the place within your lymphatic system where your immune cells hang out the most, or where you find most of your immune cells are scattered around the body in all of your lymph nodes. So know those two functions of lymph nodes. Yeah, and I put arrows here, but this part isn't important. Blood will flow in through these afferent lymphatic vesicles, through germinal centers in the cortex, then through the medulla, and out through these efferent lymphatic vessels. The spleen, I don't want to spend a lot of time on the spleen. You should just know that the spleen is what removes dead red blood cells from the circulation. It does other things, but that's really all we care about because we don't have time to know everything. The thymus also we're not going to spend a lot of time talking about. It is most prominent in infants. Well, not prominent. It exists in infants, and it grows throughout your early childhood and then starts shrinking in adolescence. And what it is really is the quality control check, or the quality control stop for your lymphocytes. So before your T cells or thymic cells are released into the bloodstream, they are tested in the thymus, and if they don't pass these two tests in the thymus that we'll talk about later, they are told to disassemble themselves, cell suicide or programmed cell death. So they off themselves in the thymus before they can enter circulation if they're not functioning properly. Then you have the tonsils. This, as this says here, is lymphatic tissue that you find in your pharynx or your throat. As I understand it, what happens here is what's this part here, right? Your immune system is sampling environmental pathogens. So if you eat something and it has bacteria or viruses in it, and your stomach acid kills those viruses, and the mucous membrane in your mouth keeps the viruses or bacteria from infecting you, you have ingested microbes that are in your environment without getting infected. And what your tonsils do is take little samples of whatever's passing through your throat so that your immune system can be exposed to the everyday pathogens that are in your environment, and you could have an exposure of your immune system that doesn't result in an infection, so that you can develop immunity to things without having to actually be infected with them. So it's sort of like a natural way of getting vaccinated, only obviously not as effective as vaccines, because we still need vaccines. Next up is the appendix. It is right here, little finger-like extension that hangs off of that first pouch of your large intestine called the cecum. And you simply want to know that it is lymphoid tissue associated with your large intestine. This part right here may house good bacteria. This is what I would consider the growing scientific consensus. I don't think scientists are all in agreement yet as to the function of the appendix, but there is growing evidence that what it might do is be a reservoir for good bacteria, because we're all covered with bacteria inside and out, and our bodies have evolved with this bacteria. So we do better when we have the proper microorganisms living in and on us. But occasionally you get something like the flu, right? And certain strains of the flu are going to cause your colon to empty out. Their blue emptying of the colon. So that empties out the colon. It disrupts greatly the bacterial flora, the populations in your colon. And part of the process of recovering from flu or any bout of gastrointestinal distress is to repopulate your colon with the bacteria that's supposed to be there. And it's thought that the appendix serves as a little reservoir because it's not in the main channel, so it doesn't get emptied out if you have a bout of diarrhea, and then you can then repopulate from your appendix the healthy bacteria. Sorry, I'm just having fun drawing now. So that's the appendix. Then diffuse lymphatic tissues you just want to be aware of. Again, MALT stands for mucosa-associated lymphatic tissue. So this is going to be respiratory tract, GI tract. I don't know if there's any in your urogenital tract because it's not nearly as likely to have microbes in it as your respiratory and digestive tract. And then Peyer's patches are a specific example of MALT. They can be seen down here where the arrows are pointing. And again, it's just lymphatic nodules that are part of the intestine. So you simply want to know that that's what Peyer's patches are. So now we're getting into the immune system. I used to do virus versus bacteria at the end if we have time. But I moved it forward because it's good to know about viruses versus bacteria, and it's definitely good to know about viruses given that we're in the middle of a global pandemic. And I'm assuming I'm going to use this recording for future classes. So if it's 2 or 3 years after the pandemic, you should know that I was recording this while everybody was working from home and wondering when we were going to be able to go out to eat again. So at any rate, it is important to understand the difference between a virus and a bacteria because we treat them differently, right? A bacterium is, as it says there, a living complex cell. It has the ability to live and divide outside the human body. It has in its cytoplasm, ribosomes, it has DNA, but it also has just all these other enzymes. It is almost as complicated, maybe even in some ways more complicated than a human cell. But all of the processes that go on to support life are going on inside of the cytoplasm. So all sorts of synthesis reactions, degradation reactions, all kinds of biochemistry, breaking down food, generating ATP, pretty much anything that your cell does, a bacterial cell does. And then they make you sick, generally here by damaging the tissues that they're in, and it might be just by eating them, or it might be because they secrete toxins that end up causing tissue damage or cell death. And a lot of times it's just their metabolic byproducts that they release that end up damaging your tissue. So what we normally treat bacteria with are antibiotics. These are small molecules, drugs, that interfere with the machinery of a bacteria. Some of the most popular ones slow down bacterial protein synthesis because their ribosomes are different than our ribosomes. So you can have drugs that mess up the biochemistry of a bacteria that don't mess up our biochemistry because our enzymes are so different. So completely different structures even though they have similar functions. Uh, a virus doesn't have nearly as many moving parts. Right, so any bacteria has thousands of different proteins floating around in the cytoplasm. You can count on two hands the number of molecules in an individual virus. So this is, I went out and got our friend COVID. This is presumably what COVID-19 looks like. Inside of it is simply RNA plus a protein scaffold to help organize the RNA. But that's it. There's no cytoplasm, there's no enzymes, no ribosomes, no metabolism of any sort takes place inside of this virus. It is not really alive. It is a genome with just enough stuff around it to protect it and make it infectious. So this virus has a phospholipid bilayer similar to ours with some proteins inserted in it. It is these pink spike proteins here on the COVID virus that you are going to see later is responsible for it entering into our cells. It is basically a key that unlocks a door on your cell so this virus can get inside. Then once it gets inside, it hijacks your normal intracellular machinery to make more viruses. So it relies on your cell's ability to perform metabolism and hijacks that and turns your cell into a virus factory. And then eventually that cell is going to die. And that's where the big damage of viruses comes from. Excuse me, because when one of your cells dies unexpectedly, they just spill their guts. And anytime a cell ruptures, your immune system reacts to that quite strongly because it takes it as a physical insult and a possible route for further infection. So you get a lot of inflammation. And it's really your body's response to the virus that causes all of the symptoms. So you get lots of inflammation in your lungs, but all of those inflammatory chemicals spread throughout your body and you get inflammatory responses in other organs, which is why, a flu, which might be infecting your lungs, could cause diarrhea or vomiting because your immune response to the virus spreads beyond the location of the actual viral infection. So that's COVID. Moving right along, this is not important, so we'll skip it. And then this I just totally stole. A couple of these things I stole. It's a pandemic. I'm not worrying about copyright protection. They can sue me later. But this is the life cycle of a coronavirus. And presumably it's the current coronavirus. No, this is different. This is SARS. Severe acute respiratory syndrome coronavirus 2. But very similar to COVID-19. I forget, it might be the same thing. They've got all these different names for it. But so again, what this virus does, it's got RNA on the inside, and this spike protein here interacts with ACE, angiotensin converting enzyme. So this is kind of like receptor-mediated endocytosis. What it does is it forms some sort of functional attachment with this receptor, which then triggers the cell to bring the virus inside of it. So it endocytoses the virus, not phagocytosis. It's not going to digest it. It just brings it in, and then the virus uncoats. So the vesicle opens up, the virus kind of falls apart, and that frees up the viral genome, which is RNA. And your cell's own ribosomes will go, oh, look, this is RNA. I'm going to make proteins from it, because that's what ribosomes do. So your cell's ribosomes get busy manufacturing all of these viral proteins here that are going to end up back in new mature viruses. One of the enzymes encoded, or one of the proteins, I should say, encoded in the original viral genome is an enzyme called replicase. What that enzyme does is make copies of the original RNA genome. And I don't know exactly how it works, because as you can see here, this is 3' to 5', and this is 5' to 3'. So there's some weird stuff going on there. But your cells, human cells, do not make copies of RNA from RNA, right? We make all of our RNA is made from a DNA template. So that replicase enzyme does not exist in human cells, and that is one of the few targets that we can use drugs for to treat the virus, is to try and develop antibiotic type drugs that can interfere with the function of that enzyme and slow down the replication of the viral genome, so that hopefully your immune system can kill this cell before it churns out too much virus over here on this side. And I think this enzyme is what Resveratrol, or I'm going to say it wrong. It begins with R, it has a V in it somewhere, and it ends with a DIR. That is one of the more promising antiviral medication that is in the pipeline. And by now, I think most people have given up on hydroxychloroquine because it doesn't seem to work very well against COVID, and a lot of people don't tolerate it very well. If you want to know more, you can listen to Mr. Ninja Nerd of Ninja Nerd Medicine, and he's got a whole hour description of it, and he gives a little background on, I think it's the SARS and the MERS viruses. So if you want to know way more than I could explain, because I'm not a virologist, you can watch this video. They give a disclaimer saying that they're not epidemiologists, and they say what the date is when they made the video, because our understanding of the virus is still ongoing. And I haven't watched the whole video. I don't know if they get into any of the epidemiology, right? But the numbers in terms of how fast does coronavirus spread, how many people that get it are going to get sick, like we're still getting updates on how many people get sick from it, how many people die from it, who's susceptible. So take a grain of salt if there's any information in there about, death rates and epidemiology, that's still in flux. Okay, we're 27 minutes long so far. We're going to keep going. This is a picture that I stole from the previous publisher, so don't tell them that I'm violating copyright, but I like it. It pretty much sums up the entire nervous system. So you have innate defenses and adaptive defenses. We are going to start with innate defenses. Non-specific, no memory, we will explain when we get there. And the same down here, specific and memory, we will explain. Non-specific versus specific, I think you'll see, will be pretty easy to understand, but the memory part won't make sense until we get down to how do your B cells and T cells work. So with the innate defenses, there's some old language from the previous publisher which still shows up in some of my questions about first-line defenses and second-line defenses, so you want to pay attention to that. So what we're going to do then is just go through first-line defenses, second-line defenses, talk about your innate defenses, and then adaptive, and we're going to do humoral and cellular immunity in that order. So this is a good outline slide just to start with. Oh yeah, there should be a space there. I was originally thinking about deleting that slide, so I used this slide. So this is the OpenStax books version of it. If you see here, innate, still non-specific, no memory, and it's surface protections, skin and mucous membranes, and then all the internal defenses, and then your adaptive immunity. So either one, this just is a little bit more easier to visually organize than that. So first up, innate defenses. You want to know that your first line of defenses is your simple physical barriers. These are your skin and your mucous membranes. associated with your first line of defenses are some chemical barriers built into your skin and mucous membranes. The two that I am most likely to ask questions about are the acidity. So you have what is called the acid mantle. The sebum and some of the other secretions that end up on the surface of your skin are somewhat acidic. This inhibits or discourages bacterial growth so that there's not as much bacteria living on your skin and fewer opportunistic organisms that would infect like a little cut or something. And then the other thing which is more important is to remember that the female urogenital mucosa or the vaginal mucosa is acidic, which is a very effective, very effective antimicrobial agent. I don't want to get on my soapbox, but there are a lot of products out there that somehow imply that the female reproductive tract needs a lot of maintenance, or that female hormones are just so likely to get out of balance and you need to rebalance yourself or whatever. Eat healthy, shower, all of that stuff, and your body will generally take care of itself. And women are not more likely to have hormonal imbalances than men. But whatever, our society likes women to feel bad about their bodies, so we sell them all of this crap. Sorry, I did just get on my soapbox there, but it happens to be true. Moving right along, so if your skin or mucous membranes are breached, your first line of defense, then you have your second line of defenses. And we're going to have cells and chemicals as your two different types of second line defenses. So the innate part of your immune system is the white blood cells that are not your lymphocytes, right? So your monocytes become macrophages. They eat bacteria that's not supposed to be there. Your neutrophils kill bacteria. Eosinophils, roundworms, and basophils are part of your inflammatory response. Here's where we can sort of talk about how the non-specific part of your innate defenses works. So if you are a neutrophil, let's say you're a neutrophil and your name is Stacy, I don't know, right? Stacy as a neutrophil will eat E. coli. Stacy will eat Staphylococcus aureus. Stacy will eat botulinum or Clostridium, right? So there's no specific bacterium or virus that any given neutrophil will attack. It doesn't matter. Right? Same thing with eosinophils and macrophages, right? All macrophages can eat any virus. You don't have one macrophage that phagocytizes HIV and another macrophage that would phagocytize the poliovirus or a coronavirus. It's just all non-specific. All targets are treated the same. Then there's this other cell type called natural killer cells, which, as it says down here, is easily the coolest cell in your bodies. These are cold, bad to the bone assassins. If you don't look right, they kill you. And what they do is a lot of scanning cells in your body to make sure that they have the proper surface receptors or the proper proteins on their surface. A lot of times, if a cell is infected with a virus or if it has cancer, its physiology changes, and that can show on the surface especially if it's undergoing physiological stress from a viral infection. Even if the natural killer cell can't detect the virus, it does know when a cell is sick and it will just kill it. We don't know really how well natural killer cells are at killing cancer cells, because we're only aware of it when they miss one and you develop cancer. We don't know how many times your immune system wipes out a cancer cell with no problems because we're never aware of that. But that is one of the things that your natural killer cells do, and they're just awesome to think about. Then we're on to chemicals, also part of your second line of defenses. Your mucous membranes or skin having been breached. You have what are called interferons. So these are chemicals, not proteins, but some sort of chemical that infected cells release to alert their neighbors. So this cell here is infected. This one's a goner. It's going to die. It's releasing viral particles, but it's also releasing interferons. So the interferons can activate immune cells and tell them to start fighting the virus. It can actually trick cells around it into undergoing apoptosis. So if they are infected, they will kill themselves before they can release too much virus. And if they kill themselves in an orderly fashion, the way apoptosis is, the viral particles inside of them don't escape. They stay membrane bound, and then a macrophage is going to come over and just digest everything. And then they can also, interferons can also cause potential target cells to put up some sort of defense. And this might be pulling in the receptors from the surface so that the virus can't get in, or, as it says here, reducing protein synthesis so that if they do end up getting infected, they're not making viral particles as fast. So that's your interferons. Then we have complement. Complement is one of my favorite things ever. It is hard to explain, but it is totally cool. So the first thing you want to know is, what is complement? As it says up here, it is 20 proteins that circulate in an inactive form. So you basically want to think of them as a football team, like the offensive side of a football team, waiting for somebody to call a huddle, or waiting for the center to snap the ball. Like, they're all ready to go, but something has to happen physically that is going to organize them, so then they're going to do what they do. And what this really means is they need something to stick to. So you're going to see that there are two ways to activate complement. What we have here is one way, which is through antibodies. So the antibodies here, these two blue things, stick to a protein on the surface of this bacterium, and then the complement, this C1 protein, sticks to that. And this is sort of the first domino. Once you get complement sticking to something, all hell breaks loose. So then, again, whoops, let me go back real quick. So the two ways to activate it and the three different functions. Here are the two ways to activate it. The one we already talked about, the classical pathway, is when complement bumps into antibodies marking the surface of a pathogen, and that sets in motion this big biochemical cascade that is complement. The alternative pathway is other proteins from the bacteria that other complement molecules might bump into. And if that sounds vague, it's because it's supposed to be vague, because we don't want to know the details. So complement can be activated by antibodies stuck to a bacterium, or complement can sometimes stick to the bacterial proteins themselves and become active. Then, once that happens, from this line here down, this cascade of chemical reactions or enzymatic interactions proceeds very rapidly once the complement is activated. Because you want to remember there's a lot of it, and it's always floating around. So it's like a big team of killers that's just waiting for the word, and then once they get the word, they all put their plan in action and run it through to the end. And this is hard to see, I know, but this is like a lot of other, like the angiotensin pathway that we talked about, and maybe kind of like the proenzymes, where you have one enzyme activates another enzyme, which activates another enzyme, and they like to chew each other apart and produce little particles. So where are we? Right here, this protein right here is going to get split in half. One half of this protein, C4b or C3b, don't worry about the numbers, it's a little protein fragment that is good at sticking to the surface of the bacteria. It also happens that your macrophages have receptors on their surface that like to stick to the complement. So when the complement sticks to the surface of the bacterium, it makes it easier for the macrophage to physically grab onto the bacterium and then pull the bacterium inside of it, phagocytose it, and digest it. So phagocytosis is not like sneaking up behind somebody and throwing your arms around them before they realize you're there. It's more you grab hold of the bacterium and pull it in towards you. And the complement just gives the macrophage something to grab hold of. So that's opsonization, coating the bacteria with stuff that phagocytes like to latch onto. The other thing that happens is enhancing inflammation. So again, this C3 protein here is going to get cleaved. This C3A causes inflammation, so it's like histamine or any other inflammatory chemical. When this gets produced, white blood cells show up. So you're basically raising the alarm and letting the rest of the immune system know that you've found a protein to stick to. Then all of this craziness happens. So you have all of these other proteins that were just waiting around, presumably for the arrival of C5b. Right? This is what we were waiting here to show up there, or maybe it's C5a, but I don't see C5a. But once you have this protein, all of these other proteins use that as a trigger, and they self-assemble and form a pore that puts a great big indiscriminate hole in the bacterium. So now it doesn't matter whether you've got sodium channels or aquaporins or this pump or that pump or this carrier. You've just got gaping holes in your membrane that let anything in and anything out. And once you've lost the ability to control what's in and what's out of you as a cell, you are not long for this world. So they end up getting have all these holes poked in them, and then they die. And because they're coated with delicious stuff that the macrophages like, the macrophages come along and clean up all the dead bacterial corpses. And it's just wonderful, beautiful carnage. I am not going to play this video. You can watch this video if you like to. It goes into a little bit more detail than I did. So I will say, just to review what you want to know, what is complement? Complement is a group of circulating proteins waiting for something to stick to. How is it activated? It can be activated by antibodies stuck to a bacterium, or it can be activated by the bacterium itself. What does it do? It does three different things. It opsonizes bacteria, making it easier for phagocytes to eat them. It causes inflammation, which is calling for backup. More immune cells are going to show up. And it forms this membrane attack complex, which pokes holes in the bacteria and kills them. So that's in summary what you want to know about complement. All of the details you don't need to understand, although I think they're fun. Mostly because if you don't have to know all of the details of a complicated process, it's just fun to enjoy the complexity. It's kind of like listening to classical music knowing you're never going to have to master the violin. Next up, the inflammatory response, or why some inflammation is a good thing. So the reason why we have inflammation in the right places, at the right times, and to the right extent, as long as you don't have too much inflammation, right? Inflammation is a good thing. And it's really items 2 and 3 here that I focus in on, right? So inflammation means you're going to attract a lot of white blood cells. Well, white blood cells kill pathogens and clean up debris, so that's good. And right, none of us like the heat and swelling that comes along with inflammation. But that heat and swelling is lots of blood, which brings all of the nutrients and raw materials that your body is going to need to fight the infection and then rebuild the tissue. And the heat is just a byproduct of all of that metabolism. So a little bit of inflammation is a good thing. I do think that inflammation is one of the ways in which our bodies could be improved. A lot of people like to think that, our body is almost too perfect a machine to believe could be sort of developed or evolved by a process like evolution. But if you really think about all of the damage that inflammation does, so again, if you have the flu, there's a virus that's in your lungs, but you might be throwing up and spending lots of time on the can with a nasty bout of diarrhea because you've got systemic inflammation that's causing your bowels to go crazy on you just because you have a virus that's in your lungs. So there's a lot about our inflammatory responses that could be improved. So at any rate, we're still assuming that most of the time inflammation is a positive adaptation, even though we seem to spend a lot of our times trying to reduce it, taking ibuprofen for that nagging sports injury or whatever. At any rate, this is why some inflammation is good. This is how it works. So the first thing you want to know is what can trigger inflammation. It can be the presence of infectious microbes, either the microbes themselves or metabolic byproducts. Your white blood cells, obviously, histamines released from basophils and other white blood cells can cause inflammation if they detect infection. But sometimes it's just tissue damage, right? You can go in for surgery, have a perfect, almost sterile antiseptic surgical procedure, no post-surgical infection, but you're still going to get some redness and swelling and inflammation because anytime a cell gets broken open when you make an incision, you're cutting through lots of cells. Anytime a cell spills its guts, your body responds by generating inflammation because that's how your body gets lots of healing nutrients and repairing white blood cells to a site of injury. So you want to know the possible sources of inflammatory chemicals, and then I put the inflammatory chemicals into two different categories. One category is one chemical unto itself, leukocytosis inducing factor. We want to know that inflammatory chemical by name because it causes leukocytosis, or the production of lots of white blood cells. As far as I know, it is the primary most important inflammatory chemical that does that, so we want to be aware of that. Then you have histamine, complement, interleukins, prostaglandins, a whole array of other immune modulatory molecules that cause inflammation. So I just group them all together and I call them histamine and the rest of the stuff because they all have more general effects, which is these two things here. So they attract more white blood cells and they increase blood flow and capillary permeability. So moving forward, right, if you're sending more blood into a blood vessel because of inflammation and you're making that blood vessel even leakier, so you're going to have higher pressure here in a leaky blood vessel, lots of fluid is going to leak out. That fluid we call edema. That's where the swelling comes from. And it's generally a good thing because it's got all of the stuff in it that you need to fight an infection and repair the damage. So again, clotting proteins, repair enzymes, glucose, antibodies, all of the raw materials and fuel that you need to clean up and repair the damage. Fever, this one is also interesting. So this is your body's response to an infection. So where is it? Leukocytes, some of your white blood cells and macrophages, when they are fighting a virus, will secrete, I don't know if they're proteins or just chemicals called pyrogens, and the pyrogens are made by your body and cause your body's temperature to go up. Presumably this makes it a little less habitable a place for viruses and bacteria, but this is another case where if your body overreacts, you know, a little bit of fever might help you fight an infection, but a lot of fever just ends up making you feel worse and can cause brain damage and all sorts of other stuff. So our bodies work well, but they are far from perfect, and our immune system definitely tends to overreact to some things and not react enough to other things. I am going to stop there. That is almost an hour. Adaptive defenses is kind of complicated. So this is a good stopping point, and then the next video we'll pick up with adaptive defenses.